Histology is the study of the microscopic structure of tissues, and it plays a vital role in understanding the complexity of the human body. In this script, we will delve into the fascinating world of histology, focusing specifically on muscle tissue. We will explore various topics, ranging from the types of muscle tissue to the molecular mechanisms underlying muscle fatigue. By the end of this script, you will have a comprehensive understanding of muscle histology and its significance in the field of medicine. Skeletal muscle tissue is responsible for voluntary movement of the body. It is characterized by long, multinucleated fibers with striations. Cardiac muscle tissue makes up the heart and has branching fibers with intercalated discs for coordinated contractions. Smooth muscle tissue lacks striations and has spindle-shaped cells that organize into sheets or bundles. Each skeletal muscle fiber is composed of myofibrils, which are arranged longitudinally. Myofibrils contain repeating units called sarcomeres, the functional units of contraction. Sarcomeres consist of thick and thin protein filaments, which are responsible for muscle contraction. Thick filaments are formed by myosin, while thin filaments consist of actin, tropomyosin, and troponin. The organization of these filaments within sarcomeres results in the characteristic striations observed in skeletal muscle tissue. The sliding filament theory explains how muscle contraction occurs at the molecular level. According to this theory, during contraction, the thin filaments slide past the thick filaments, causing the overlapping actin and myosin filaments to generate force. This process is regulated by calcium ions, which bind to troponin, leading to a conformational change and exposing the binding sites on actin. Myosin heads then bind to actin, undergo a power stroke, and subsequently detach, allowing for the progressive sliding of the filaments. Neuromuscular junctions are specialized synapses between motor neurons and skeletal muscle fibers. These junctions play a critical role in muscle contraction by transmitting signals from the nervous system to the muscle. When a motor neuron releases acetylcholine, it binds to receptors on the muscle fiber membrane, leading to depolarization and the generation of an action potential. This action potential propagates along the muscle cell membrane, ultimately triggering the release of calcium ions from the sarcoplasmic reticulum. Excitation-contraction coupling is the process by which an action potential results in muscle fiber contraction. Once calcium ions are released into the sarcoplasm, they bind to troponin, causing a conformational change in the exposure of myosin binding sites on actin. Subsequently, myosin heads bind to actin, forming cross bridges. The myosin heads undergo repeated cycles of binding, power stroke, and detachment, leading to the sliding of the thin filaments past the thick filaments. Motor units are functional units consisting of a motor neuron and the muscle fibers it innervates. The number of muscle fibers innervated by a motor neuron varies, depending on the precision of movement required in a particular muscle. Muscles involved in fine movements, such as those in the fingers, have smaller motor units, while those involved in gross movements, such as the quadriceps, have larger motor units. The activation of motor units is crucial for coordinated muscle contraction. The cardiac conduction system consists of specialized cardiac muscle cells that initiate and propagate electrical impulses, ensuring the coordinated contraction of the heart. It includes the sinoatrial, SA, node, atrioventricular, AV, node, bundle of His, bundle branches, and Purkinje fibers. The SA node acts as the natural pacemaker, while the AV node delays the electrical signal to allow for complete ventricular filling. The subsequent conduction through the bundle of His and Purkinje fibers results in synchronized ventricular contraction. The contraction of cardiac muscle is similar to skeletal muscle contraction, but with some key differences. While calcium ions play a vital role in initiating contraction in both muscle types, in cardiac muscle, calcium ions primarily come from extracellular sources. Additionally, cardiac muscle has a longer contraction duration and is capable of sustained contractions due to the presence of calcium-induced calcium release mechanisms. This enables the heart to efficiently pump blood throughout the body. Smooth muscle cells exhibit spontaneous contractile activity, allowing them to maintain muscle tone, vital for various physiological processes. The tone of smooth muscle is regulated by the autonomic nervous system and various local factors, such as changes in intracellular calcium levels. Smooth muscles respond to neurotransmitters, hormones, and mechanical stimuli, which can either stimulate or inhibit contraction, depending on the specific signaling pathways involved. This tight regulation ensures proper function and control of smooth muscle. Muscle fiber hypertrophy refers to an increase in the size of muscle fibers due to increased protein synthesis. This occurs in response to mechanical stress, such as resistance training. Hypertrophy results in an increase in muscle strength and volume. On the other hand, muscle fiber atrophy refers to a decrease in the size of muscle fibers, which often accompanies prolonged inactivity or disuse. Understanding the mechanisms underlying hypertrophy and atrophy is crucial in the context of muscle building and rehabilitation. Muscle disorders encompass a wide range of conditions, including muscular dystrophies, myopathies, and neuromuscular diseases. These disorders can arise from genetic mutations, autoimmune processes, inflammation, or metabolic abnormalities. They often result in muscle weakness, impaired coordination, and progressive degeneration of muscle tissue. Diagnosis and treatment strategies for these disorders require a comprehensive understanding of muscle histology and the underlying pathophysiology. Muscle fatigue is the temporary inability of muscle fibers to maintain or generate force. It is influenced by complex molecular mechanisms, including the depletion of energy stores, the buildup of metabolic byproducts such as lactate, alterations in calcium handling, and disruptions in the excitation-contraction coupling process. Understanding the molecular basis of muscle fatigue is vital for athletes, patients with chronic fatigue, and those with certain medical conditions where fatigue is a prominent symptom. Through histology, we can explore the complex world of muscle tissue, understanding its structure and function. From the organization of skeletal muscle fibers to the mechanisms of muscle fatigue, this knowledge helps us diagnose and treat muscle-related disorders, improving patients' lives.